In this next section about the immune system, we're going to look at specific adaptive immunity. So in our last video, we talked about innate immunity, which was non-specific, the immune system that we are born with, and that is going to behave the same way, really no matter what type of pathogen or injury that we're exposed to. So when we start looking at adaptive immunity, we're going to see two very different qualities: specificity and memory. So specificity is when the cells of the immune system are going to target very specific pathogens. And memory is the fact that we have cells of the immune system that remember our previous exposures and then can adapt and respond very, very quickly to those. So specificity and memory are achieved by being able to program immune cells to respond rapidly to a subsequent infection of the same or very similar pathogen. So when we are exposed to a pathogen for the very first time, that's called the primary response. So the immune system is going to launch a response. We'll have basically a two-armed approach. We'll have the responses from our innate immunity, and then we have the responses coming from our adaptive immunity that are going to remember that pathogen. In the secondary response, the next time that we're exposed to that particular pathogen, we're going to have a faster and stronger response. Okay, and that again is due to the memory of those. That secondary response is going to be very specific to that particular pathogen. We looked at a lot of different cells that were responsible for innate immunity, but when it comes to specific immunity, we're looking at our lymphocytes, specifically B lymphocytes or B cells and T lymphocytes or T cells. B cells and T cells arise from the same stem cell, but they take a different pathway to maturation, and so they behave differently. B cells mature in the bone marrow, and that's where their name comes from, and they are responsible for the production of antibodies, sometimes called immunoglobulins. This arm of the immune system is called humoral or antibody-mediated immunity. T cells mature in the thymus, and that's where their name comes from. T cells are basically the orchestrator. They are responsible for both the innate and adaptive immune responses, and they target infected cells. They are responsible for cell-mediated or cellular immunity. They do not produce antibodies. Only the B cells produce antibodies. In order to launch a response against an invader, regardless of what that is, we have to be exposed to these antigens. Antigens are pathogen-specific molecular structures. They're kind of responsible to those PAMPs that we looked at in the last video. PAMPs are found on numerous different pathogens. Antigens are specific to a particular pathogen. Antigen was originally described or used to describe molecules that stimulated the production of antibodies, and that's where their name comes from, antibody generating. But we realize now that antigens are not just related to humoral or antibody-mediated immunity. And so sometimes they're referred to as immunogens because they really stimulate a response by the entire immune system and not just the B cells, not just those antibody secreting cells. Antigens can be any number of things. They can be carbohydrates, they can be lipids, they can be proteins, they can be glycolipids or glycoproteins. So they can be a lot of different structures, and they vary in how they're able to stimulate our adaptive immune defense, as well as are they more going to trigger a humoral or antibody mediated response, or is it going to be a cellular response? More complex molecules are more effective as antigens. And one reason for that is that the antibodies and the T cells don't recognize an entire antigen, but rather a specific section of the antigen called an epitope. And one antigen, especially if it's larger, can contain multiple epitopes, which means that we can have different antibodies that bind to those epitopes. So we can have several different antibodies that basically act against the same antigen. The antigen size is also important in its antigenic potential. Some molecules are too small to be antigenic by themselves. They're called haptens, and they're basically free epitopes. And for a hapten, in order to trigger an immune response, basically for it to become antigenic, it has to attach to a larger molecule. Haptens are responsible primarily for allergic responses. For instance, people who are allergic to poison ivy are actually allergic to the oil called urushiol, and this is considered a hapten. So antibodies or immunoglobulins are glycoproteins. So they're composed of carbohydrate and proteins. They float around in the blood as well as within the fluids in tissues. And they kind of make this roughly Y-shaped structure here. And on the end of each of these little chains here will be the spot where they can bind to the antigen. Now, there are 5 classes of antibodies, G, M, A, D, and E, and they're going to be a little bit different in terms of how abundant they are, their specific arrangement, where they act, what they do, and how big they are. So IgG is the most abundant in the blood, about 80% of our total antibodies, but it can get into tissue spaces. It also crosses the placental barrier. So when we talk about, the baby being able to receive antibodies from its mother, we're referring to these IgG antibodies. It's also the most versatile in terms of the different pathogens that it can interact with. IgM is a little bit different. It can perform some of the same functions of IgG, but it's the first antibody that's going to be produced and secreted by the B cells in both the primary and secondary immune responses. So we can look for IgM in the blood because it's going to be elevated during active or recent infections. IgA is about 13% of total serum antibody. It is found in the mucous membranes, breast milk, tears, and saliva. Because it's found associated with those mucous membranes, it can trap pathogens in mucus. IgD is similar to IgM, but it's not secreted by B cells, and we only have trace amounts of it. And then IgE is the least abundant. It's going to bind to basophils and mast cells, and basically is triggering defense against parasites and also functioning in allergic reactions. So you've got this table here that kind of summarizes those. So when we look at these antibodies and their interaction with the antigens, they have several different ways that they can defend us against pathogens. They can neutralize the pathogens. They can opsonize the pathogens and allow them to be phagocytized. They can agglutinate or make them clump together. They can activate that complement cascade that we mentioned in the previous video. And they can trigger the cell-mediated cytotoxicity, basically, triggering that cell to be destroyed. So neutralization is when antibodies bind to epitopes, preventing them to attach to cells. So if they can't get into the cell, especially if we're talking about those obligate intracellular parasites, if they can't get inside the cell, they can't cause disease. So these are effective against those types of pathogens as well as viruses. Opsonization we talked a little bit about in the last video, but this is basically when we coat a pathogen with some sort of molecule that allows it to be able to be marked for phagocytosis. IgG antibodies serve as these opsonins that allow it to be phagocytized. Agglutination is when we get the cross-linking of pathogens to create these large aggregates or large clumps of pathogens. It's easier for the kidneys and for the spleen and even the lymph nodes to be able to remove those from the body. The complement cascade, so antibodies can activate or trigger the complement cascade. And then the antibody-dependent cell-mediated cytotoxicity, which is kind of a mouthful, ADCC. This is when we have pathogens that are too large for our phagocytes to be able to ingest them. And so one of the cells that we see do this, we talked about, were the natural killer cells, but macrophages and eosinophils can also do this. But basically what happens is an IgG antibody is going to bind to a large pathogen, and the effector cell doesn't ingest them altogether, but instead secretes these cytotoxins. So these chemicals that are toxic to the cell, and it kills the cell that way. So basically the antibodies are marking that cell for destruction. And these natural killer cells are going to have a receptor that's going to bind to those antibodies to allow that to happen. So when we were talking about innate or nonspecific immunity, we talked about the major histocompatibility complex molecules, or MHC molecules. These are on the surface of healthy cells, basically identifying them to the immune system as normal, as self, so that our immune system doesn't start to attack them. But MHC molecules also are important in presentation of foreign antigens. So they can be used to tell the immune system, hey, something is wrong here. And this activates T cells, which then activates the entire adaptive immune system. So the MHC, major histocompatibility complex, is a collection of genes. It codes for those MHC molecules to be found on all nucleated cells. And in humans, we also call these HLA, human leukocyte antigen genes. So we have two classes of these, MHC1, MHC2. MHC1 are found, again, on all nucleated cells. They present normal self-antigens as well as abnormal or non-self pathogens to effector T cells. The MHC2 molecules are only found on macrophages, dendritic cells, and B cells. They only present abnormal antigens. Okay, so MHC1 is found on all nucleated cells, and they can present both normal and abnormal. MHC2 molecules only found on macrophages, dendritic cells, and B cells. They only present abnormal antigens for activation of T cells. We also have other types of cells called APCs, or antigen presenting cells. And these are found, or these cells are the macrophages, dendritic cells, and B cells. And They're different, but they do play similar roles in adaptive immunity. Macrophages and dendritic cells, they are phagocytes. They are part of our innate immune system. B cells are not phagocytes. Remember, they produce antibodies. So the macrophages and dendritic cells can directly attack the cells, directly phagocytize them. But B cells secrete antibodies, alerting the rest of the immune system that there is an issue. So looking a little bit more specifically at T cells, this is a scanning electron micrograph of a T cell. You see it's got these little spike-like structures all over it, basically increasing its surface area to allow it to interact with other cell types a little bit more readily. So T cells, again, they are maturing in the thymus. And I'm not going to go through this, but there is a process for how they mature and how they are formed. But T cells are classified into 3 different classes: helper Ts, regulatory Ts, and cytotoxic Ts. And they're differentiated based on what type of surface molecules they have, how they're activated, and basically what they do. So they've got these CD molecules, or cluster of differentiation molecules. And this table kind of summarizes what they do. So helper Ts are activated by antigen presenting cells associated with those MHC class 2s. They basically start the whole thing. They orchestrate, they direct both the humoral or antibody mediated as well as the cell mediated immunity. And they also are responsible for, in part, some of the innate immunity, because they help activate the macrophages and natural killer cells as well. Regulatory T cells, also activated by the same type of molecules as helper Ts, but they help prevent autoimmune responses. So basically, regulatory T cells are helping to regulate the immune system, prevent it from getting way out of hand. And then the cytotoxic T cells associated with APCs or MHC1, they directly destroy cells infected with intracellular pathogens. So this is where we get that term cellular immunity or cell-mediated immunity. So the rest of this section just kind of goes through a little bit more detail about the T cells. So I'm going to skip through that and jump over here to our B cells. So B cells participate in antibody-mediated or humoral immunity. B cells are going to have antigen-specific receptors on them. They do rely on T cells to become activated, but they can become activated on their own. But we really need that double arm of the B cells and the T cells to produce a proper immune response. So when we look at B cells undergo what's called clonal selection, and these differentiate into memory B cells and plasma cells. Memory B cells are those that remember. Okay, so they're responsible for remembering the pathogens that we've come in contact with, or the antigens we've come in contact with. Plasma cells, their sole purpose is to secrete antibody. So we have kind of this double arm approach where we have these memory B cells that remember and can launch that response very quickly. And we get these plasma cells that their sole purpose is just crank out hundreds and thousands of those antibodies that can attack that particular antigen. So the T cell dependent activation of B cells is important in both the primary and secondary responses. So if we kind of look at what happens, we have the primary antigen exposure. So we're exposed to something for the very first time. After about 10 days, our IgM molecules peak, or about 10 to 15 days. And then at about 20 days, our IgG peaks, okay? And so this little period here is called the lag period. Well, now when we get exposed to that again, okay, we don't have as long of a lag period. We have the IgM response stays about the same concentration, but occurs much quicker. But notice the IgG response. First of all, it's a lot quicker, but it also is, we produce much, much more of those IgG molecules. So the levels of the specific antibody then are going to also remain elevated for a longer period of time. So the last section here in chapter 18 talks about vaccines. And so vaccines basically are our primary exposure. So the immune system is learning these diseases so that if we are ever exposed to them, we launch that response. So there are different types of adaptive immunity or specific immunity. We have passive and active, and then artificial and natural. So here are the examples. So passive refers to receiving antibodies that have already formed. And active refers to your body is actually producing the antibodies itself. Natural means it happens in the course of life. Artificial means that you're injected with it. So natural passive would be something like breast milk or placenta. The baby is not making its own antibodies. It is receiving antibodies that have already been produced by the mother, but it's in the normal course of everyday life. Artificial passive is when a person is given an IgG or an IgM injection. Those antibodies have been produced by another person or by an animal. Natural active is when we contracted a disease and we recover, and then we have made antibodies against it. So, you know, before the chickenpox vaccine became available, I remember my entire second grade class had the chickenpox. So that would be natural active because we came into contact with it just in the course of everyday life, and we produced our own antibodies against it. Now, if we look at chickenpox immunity, it's artificial active because children are receiving the vaccine against the chickenpox virus, the varicella zoster virus. So it's artificial in that it's in a vaccine, but it's active because once the body is exposed to that pathogen, to that virus, it produces its own antibodies. So then we get to herd immunity. This has been on the news a lot lately in response to COVID. So herd immunity is when enough people have been vaccinated or have contracted a disease that they're basically able to protect the entire population against it. It basically refers to the fact that there's going to be too few susceptible individuals for the disease to spread effectively. And this is what happens when we attempt to, you know, wipe out a disease like smallpox. You know, most people got the vaccine, but even those who didn't were protected by those who did, and we were eventually able to wipe it out. And it's the same thing with polio, and really what we're attempting to do with, you know, measles and mumps and rubella as well. So then we get into kind of the history of the development of vaccines and then the different types of vaccines. So with vaccines, we have live attenuated. Basically, these are weakened. They produce long-lasting immunity. They produce both cellular and humoral immunity. And so a lot of these are virus. These are effective against viruses. And so when we say live, remember, viruses aren't alive. What we're referring to is that it is the entire pathogen, but it's just been weakened. Inactivated means that it's the entire pathogen that has been killed or inactivated. They're easily stored. There's no risk of a severe infection. It produces weaker immunity though, and there are more boosters that are required for this. So things like the flu. Subunit are pieces. You're basically given the antigen for it. Multiple doses are required for these as well. These are things like hepatitis B, meningitis, whooping cough. Toxoid is when you're injected with the inactivated toxin. So things like diphtheria, pertussis, and tetanus are also toxoid. And then conjugate is when the capsule is conjugated to a protein. These are pretty expensive. And these are things like some of the meningitis vaccines are conjugate. So you've heard the meningococcal conjugate vaccine, or the MCV, or the pneumococcal conjugate vaccine, or PCV. These organisms produce capsules. These are, bacteria, they produce capsules. And so if we receive immunity against that capsule, then that basically allows us to recognize the capsule and clear that disease from the body. We're also working on, DNA and RNA vaccines. And of course, the COVID vaccine is an RNA vaccine. So it is fairly new technology, something we've been working on for a long time, but relatively new. It's really the first wide scale RNA vaccine that we've had. So that brings us to the end of the immune system.